Okay, transcription 2. All right, so as I said, we'll make this kind of as a separate video just in case the kind of exons and the introns is an area that you were a wee bit unsure of. Okay, so as I said, when we copy, so the area that we're basically transcribing, approximately about 8,000 nucleotides, okay, is basically copied. But when we actually make that protein, there's only about 1,200 of them required. So there's large sections of this molecule that we can basically discard that we no longer need, okay? So when we talk about introns and exons, we're talking about the primary transcript, because at the end, once we've removed the non-coding sections, that therefore becomes our mature transcript that is ready to go and travel to the ribosome, okay? So the primary transcript, okay, this is containing exons, okay, and introns. So these are our coding sections, so our coding areas, okay. It's going to code for a protein, and these are our non-coding areas. Okay, so if we go back to our kind of diagram, let's see. So let's make a messenger RNA. Right, perfect. Okay, so here we have, let's keep it nice and easy for us. I think I'm definitely going to have to buy new pens. That's actually exciting. That's how boring life is at the moment for us all. So you, okay, so this is our primary transcript. This is what we've just copied from our open section of DNA, okay? And then what we're going to do is I'm going to call, so I'm going to say that this, so these three nucleotides, this triplet here, this codon, remember, I'm going to say is an exon, okay? And then I'm going to say that this one here is an intron, this one here is an exon, and this one here is an intron. Okay, all right. Maybe should have made the molecule a wee bit longer, but we'll stick with that just now anyway, okay? So this section here and this here, okay, are non-coding. Whereas this is coding. Okay, so essentially what we're going to do is we're going to remove these sections, the introns, okay, and we're going to take our exons, okay, that has, so that would be our A, U, G, and that would be C, A, U, okay? And what we're going to do is we are going to splice them together, okay? And this is what we call RNA splicing. It's when we remove the non-coding sections, the introns, and we splice, we join together the coding sections, okay? So we take our molecule, which is going to end up, 1, 2, 3, okay, which ends up looking like C, A, U. So this, these two exon, these coding sections put together creates my mature transcript. And again, this is just me simplifying it. This is going to be a long molecule with many, many sections that they're not coded for that particular protein that we need. Okay, but this is the importance of when we talk about, like we previously said, primary transcript only codes for one protein. Okay, because we can therefore identify these different coding sections. Okay, that therefore means that we can create different mature messenger RNA transcripts. Okay, so they can be produced from the kind of same primary transcript, but it depends on what exons, the kind of order of the exons. Okay, so if we had, so for instance, say we had exon 1, exon 2, exon 3, exon 4. For us to have the different proteins, so protein 1 could just be exon 1 and 2, whereas protein 2 could be exon 1 and 3, whereas protein 3 could be exon 2 and 3. Okay, because remember, to do the sequence of these nucleotides, it gives you that code. So although we are not removing the exons, okay, we're just changing the order. Okay, so again, this is what we call, I'll squeeze it in here, alternative splicing. So we can actually get different proteins from that one gene. Okay, so different mature messenger RNA transcripts. Let me see that, okay, transcripts produced from the same primary transcript. Depending, squeeze this in here, sorry, depending on order of exons. Okay, so this mature transcript now is fully ready to rock. So that is exon, that specific mature transcript is the exact code to make a certain type of protein. And the next part of the journey is translation. So go to the ribosome. So it travels to the, can't spell ribosome, ribosome. All right.